Okay, everyone, welcome to Microbiology. This is the first of many lectures that we're going to go through on the subject, and I think it's good we just get started. So, before we really dive into our work today, I want to give you a warning about this content and the class as a whole. This is mainly it's a half online, half in person class that we're going to be working through, and there are certain expectations that come with that. You are going to be responsible for doing a lot of at home work that is on your schedule, that is something that self motivated. There will be deadlines to complete work within a certain period of time, to go through this content within a certain period of time, but there's no dedicated class time allotted to go through a lot of this lecture material. Our class time is mostly dedicated to lab. And so it will be up to you to go through and make the time to review all of this content. And so I strongly advise that you watch each of these lecture videos in full. I realize they can be long, but it must be done. Make sure to be taking careful notes throughout this class, throughout each of these lectures. And I really recommend handwriting your notes in a notebook or something similar to that, because that can be very helpful for actually learning the content and keeping it in your head as you go through the class. And then I also include a lot of supplemental content, videos, podcasts, things of that nature for each of the sections that we go through. And those you'll find on your online portal. These are not required resources unless specified. They wouldn't be in the supplemental section if they were. But I do go through and look at these resources, and I pick them and provide them for you because I think that they can be very helpful, particularly for understanding specific concepts that we talk about in these lectures. And so I do recommend taking some time to actually go and review that content, especially if you start feeling a little stuck or you start finding yourself confused on certain subjects and you see that a video in the supplemental content is on one of those subjects. So with the warning out of the way, let's get going. For a long time, humans have been thinking and asking questions, particularly about life and about the various complexities of that life. Where does life come from? What is life made of? How is life made? How does a body work? What about disease? Where does disease come from? What about reproduction? And a ton of other really broad questions, but no less important questions. And some of these questions were very influential, particularly in the early days of humans starting to explore their natural world and beginning to ask real questions and try to come up with real answers, about life. In the days where science was in its infancy, or in some cases it really didn't exist at all, people had strange answers to a lot of these questions, at least from our modern perspective. A question like, where does life come from, was a particularly relevant and important question for people because we would make a lot of observations. People would observe that in some cases it seemed like life or living things would just seem to spawn. They would arise from things that we thought were non-living. Like, for example, if you left meat out and it rotted, it spoiled and slowly rotted away, you would see that flies would start to emerge. Maggots would grow on that food, on that meat, and eventually flies would emerge as well. People used to also believe that if you left hay out in the fields for too long, that mice would be born from that hay, because they observed that mouse dens would form in these hay mounds and mice would begin to live in them. Now, something important, humans never really observed flies coming in to lay eggs. They never really observed mice in the dead of night going and making a home in a pile of hay. But without those observations, we started to kind of fill in the blanks. We started to try to use logic to the best of our ability in these early days and start to draw conclusions. And that's where this concept of spontaneous generation came from. It was the idea that life could just arise spontaneously from non-living things. At this time, it wasn't really understood that that life could only come from life. This is something that we know today, that we kind of take for granted from our very modern perspective. It seems kind of silly from our perspective to believe that a rotting piece of meat could birth maggots. A fly would have to come and lay those eggs. We know that today. Historically, people did not know that at the time. And so this idea of spontaneous generation, the theory of spontaneous generation that they had come up with, was very popular. This idea that living things could spontaneously arise from non-living things. So, of course, these major questions that we look at were asked during this time, like what an organism is made of, or how reproduction really works, or where life comes from, things like that. They were unknown at this time. They are well known now. We were dealing at this time in human history of an issue of perception, limits of our own perception, that sometimes things would happen on time scales or spatial scales, size scales really, that were too difficult for us to observe unaided. So for example, when bread would turn to mold, when bread would eventually bear mold like this, people used to believe that was just something that happened to bread, that bread would eventually give rise to fungi. The bread that was not alive, made of organic matter, would birth living fungi. But what was really happening is that we didn't, it was hard for us to see, it was virtually impossible to see microscopic spores land on this bread and then turn into fungi that began to eat the bread. Similarly, with the maggots, we never really observed these tiny flies arrive, I should say, on the meat and lay eggs. Eggs are incredibly small, and it's a momentary blip, and no one is sitting around just watching rotting meat. So these were things that people failed to observe and had to fill in the blanks for. Likewise, we spent a lot of time observing the impacts of things that were too small for us to see with the naked eye, but we had never actually seen their cause. Things like disease. For the longest time, we had no idea about what really caused disease. Nobody saw bacteria or viruses or things like that. Those are things that are too small for the human eye to see alone. Also, fermentation as a product. There was a process that happened if you wanted to make ale or wine or things like that. You could make that stuff, but nobody really understood how and why it happened. And then medicine, similarly, that people figured out how to utilize things like molds very early on to use as essentially like topical antibiotics, among other things. And at the time, it was really unsure why this worked. No one really understood that if you slapped moldy bread onto a wound, it was the antibiotics that mold was producing that was helping fighting off that bacterial infection. It was not something that was witnessable by people at this time. So this really changed when microscopes came onto the scene. So microscopes were something that were created almost on accident, honestly. There was a person named Zacharias Janssen that in 1585, he designed the first compound microscope, and he was not interested in microscopic life. He was interested in the stars, and all of these astronomers at the time were trying to create better and better telescopes to try to see further and further into space and see things that were more clear, get better, clearer images. But in the process, the funny thing is that a lot of these lenses, that if you just kind of rearrange the lenses, you could magnify incredibly small things instead of things that are incredibly far away. And so that is what kind of happened. It was an accidental discovery that the people who made the first microscopes kind of didn't really care about. Galileo is also known for creating a microscope. Very early on in the 1600s, had multiple lenses. He cared so little about it, he didn't even name it. It was other people that were observing his discovery, his creation, and gave it a name, microscopio. Later on, you had very influential people like Antoine van Leeuwenhoek. Antoine van Leeuwenhoek was one of these people that we now call fathers of microbiology. Well known for his creation of a microscope, one that was handheld. It was just single lens and a little pin that you would put a tiny sample onto, and then hold the paddle up over your eye, looking into the sun or looking into some other light source, and the light would pass through this item, this little sample, go into the lens and be magnified, and then you would be able to see it increased in magnification by the naked eye. And Leeuwenhoek, he was fascinated by all kinds of stuff. He was one of the first people to actually start putting tiny things under a microscope and care about it. So Leeuwenhoek put water samples, saliva, plaque from his own teeth, fossils, tiny pieces of plants and animals and more on this pin and looked at it under the microscope. And he also recorded a lot of what he had seen. And so we actually have drawings, simple as they are, of course, of the microscopic life that he saw. And so, this is likely he was seeing bacteria. He was probably seeing different kinds of microscopic complex life, like protozoans and things like that. But he would actually draw what he was seeing, which was very impressive at the time. Leeuwenhoek called the things that he was observing in the microscope animalcules. It was sort of a catch-all term at the time for microbial life forms. It was animals, but also bacteria, diatoms, ciliates, amoebas, all kinds of stuff. And so this concept of animalcules is older than Leeuwenhoek, but he was the first to apply it to these microorganisms that he was seeing. And this really started this new line of thought. People at the time began to become increasingly interested in Leeuwenhoek's animalcules and started to create more complex devices to learn more about them. And with the advent of the microscope, people began to think more critically about these microscopic organisms. They began to not just recognize that living things were out there that were smaller than what the naked eye could see. They were also starting to ask, what does this mean for other living things like us? It's about this time we should go to Robert Hooke, because he kind of comes next in this timeline. He's an English polymath and early naturalist who had very diverse interests, particularly in biology, but also in math. And he is credited for discovering the cell. He was one of the early adopters of the microscope after Leeuwenhoek. And began to utilize his own very primitive light microscope to look at slices, very thin slices of different organisms, particularly like plants, focusing mostly on plants. Uh, and so one of the it was his work with cork, like the plant, that he noticed that there were tiny containers that were inside the tissue of this plant. And so for this, he coined the term cell. This is a reference to these monastery living quarters that were well known at the time. That religious people would live in these cells inside of the monastery and spend all their time in there. So they looked very much like these small rooms, which Hooke attributed immediately the name. Now, this was one person's observation. It would take more time for people to start to demonstrate that this is real, that life is composed of cells. It wasn't even really something that people were thinking about at this time. But microscopes at this time, in the 1600s, were very scarce. They were very sought after. But those few people with them began to confirm Hooke's observations. And that actually included Leeuwenhoek, because they were contemporaries. They communicated together pretty regularly. And so people like Leeuwenhoek began to study and look at slices of many different organisms, plants, animals, fungi, and more, and began to notice a consistent thing. All of these living things seem to also be made of cells, at least one cell at minimum. And so more people, as we move further in time, I mean, I'm talking about like 200 years, moving 200 years into the future, steadily building on this body of research, people began to finally recognize that everything that was alive seemed to be made of cells. Again, something that we today, I think of being very obvious, but this was something that had to be learned at the time. And so fast forward to 1838, you have two very influential figures, Theodor Schwann and Matthias Schleiden. These are both animal and plant biologists, respectively. Each of them were spending a lot of their time studying the prevalence of cells in the tissues of their study organisms. So Schwann was very interested in animals. He was looking at lots of different animal tissues and finding that everywhere he looked, he would consistently find cells. Similarly, Schleiden, as a botanist, was looking mainly at the tissues of plants, and he was finding the exact same thing. So this was building this idea of what we now think of as cell theory, that all living things are made of cells. At least one major part of that theory is slowly coming together. All the while, this idea of spontaneous generation still exists and persists pretty severely at this time. The researchers who were doing this hard work, like Theodor Schwann himself, had his own unusual ideas about spontaneous generation and about the origins of cells. He thought that cells could crystallize, that they could come together and form from more primitive matters within whole organisms, and eventually become whole organisms themselves. So, just because these people were at the same time revolutionizing science doesn't mean that they weren't necessarily dated in their other major thoughts. So another scientist named Rudolf Virchow would come into the picture around this time, and eventually convinced Schwann, who was one of these major people who was kind of thinking about spontaneous generation of cells. He convinced him that cells could not arise spontaneously, because Virchow was presenting observations from red blood cells that he was getting. He was observing red blood cells in chicken embryos and was observing them be formed. And so this is one of the earliest recognitions that cells could actually divide to create new cells. Except Virchow didn't observe that. He actually stole those observations from another researcher named Robert Remak, who was a contemporary of his. So Virchow stole his research and then published it as his own, which is, it's par for the course for science of this time. Very cutthroat, very backstabby, but person that's been somewhat forgotten from history as a result. Remak. But you put all these people, all of these influential people together and their many ideas, and an idea began to form that became known as cell theory. And so cell theory now has three parts to it. Cell theory says, number one, all life is made of cells. All living things, anything that we consider to be alive, must be made of cells. That is something that we see time and time again. It has been observed consistently. Number two, the cell is the smallest unit of organization of life, meaning that a cell is alive. Anything smaller or simpler or less complex than a cell is not alive. So a protein is not alive. Your mitochondria, a part of your cell, is not alive. Things like that. And then number three, cells only arise from pre-existing cells. We today cannot get cells to arise from inorganic matter or like non-living things, right? That you have hay that is rotting in your field. It's not going to spontaneously start creating new cells that might, develop and become mice. This is actually very intricately connected to overcoming spontaneous generation at this time. That said, it still takes a long time to kind of get there. Now, at the time, Schwann and Schleiden believed this only applied to plants and animals, but we soon recognized that this was something that all life had. All life, not just complex multicellular life, but also single-celled organisms like bacteria. All forms of life all fall within this same rule set. They all follow cell theory, and that means that all living things, particularly because of cell theory tenet number 3, the idea that all cells are born from pre-existing cells, that means that all living things ultimately are linked together. Cells splitting from other cells in a continuous chain going all the way back to the dawn of life, whatever that looked like. So all living things, us, all of these creatures, and ones that are certainly not represented here, if we are alive, and we are made of cells, we are all linked by this ancestry, which is really cool. This goes to microscopic life as well. So even single-celled organisms, we can say the same thing. All of life ultimately is related. Blue whales to bacteria and us and everything in between. All living things are connected, sharing this history of life on Earth. So that is a really important aspect to keep in mind, okay? That we've all essentially evolved from one common ancestor. One with this idea of our cells dividing and dividing and dividing in this continuous line for the last 3-point-something billion years, which is pretty amazing. Now, if you have something that is not made of cells, then what is it? An important question that must thus be asked. Well, it's not alive. That goes for viruses, prions, which are a form of infectious protein, individual molecules, rocks, all these different things. If it's not made of cells, it's not alive. And so that doesn't mean that there might be a lot of similarity. There might not be a lot of similarities between these things and life. Viruses, for example, can replicate. They can evolve just like living things, but that doesn't make them alive. That makes them capable of reproduction and evolution. That's all. So that is really important to keep in mind here. Characteristics of life, when we start thinking about what all life shares, I think this is a really important thing to keep in mind. All living things, as we now know from cell theory, are made of cells. Right? We can start there. But living things also have a bunch of other qualities. They can grow and develop. They contain some kind of genetic information, something that can be replicated and passed down as an instruction on how to make that organism, and you can pass it down to your descendants. And we store that as DNA, or for some organisms, for some things, RNA. Living things can evolve, as in their lineage can change over time. A single individual like you or me, we are not evolving, but when we have children, and our children have children, and their children have children, and so on, they will not be identical to us. There will be some traits that they have acquired from their other parent, for example. And there are some situations where maybe they've accrued a mutation or two. In any case, their genetics will not be the same as ours. And so there is change over time. They also reproduce. They make more of themselves. They keep homeostasis, a really important concept. That means a constant effort to maintain the conditions for survival, both within cells and among cells. So, for example, in your body, the neurons in your brain are doing cellular homeostasis to keep themselves alive. But also, all of your cells are working together to keep your whole body alive. They're doing systemic homeostasis. Living things will also take up nutrients, remove wastes, and generate energy. They will perform metabolism, meaning that they are actively breaking molecules and building up new ones. Living things sense the world and respond to stimuli. Sometimes not through movement. A lot of living things can move, but it's not mandatory for a living thing to move. They must only be able to adapt, change their behavior to deal with things that they sense in their environment. And so microorganisms like bacteria, like fungi and algae and different things like that, they all share these qualities too, just like we do. Take, for example, this amoeba I put in the top right corner over here. This amoeba is demonstrating that it is very much alive because it is trying very hard to swallow this, it looks like some kind of a diatom cell. And it will give a lot of effort to try to do that. It's because it senses its environment. It has detected that there's a diatom and it has responded to the presence of that diatom by prioritizing trying to eat it so that it can take up nutrients and start to do metabolism. All of this in service of homeostasis. When we look at things like viruses, there's a lot of things that viruses cannot do that living things can do. So, of course, first, they're not made of cells. But they also can't grow or develop. They're sort of born as is, created as is, I should say. They don't have any ability to keep homeostasis. There's no homeostasis to keep. They don't take up nutrients. They don't remove waste. They don't generate energy. They don't do metabolism in the true sense. And they don't sense or respond to stimuli really either. They do have genetic information. They do have the ability to evolve. And they do have the ability to reproduce, although Reproduction is something that they make a cell do for them. When a cell is infected with a virus, it hijacks that cell and it turns it into a factory that makes viral particles. So this reproduction isn't even something that the virus itself is really doing. It's not autonomous. Instead, it's completely dependent on its host. So this is why we don't call viruses living things. People can debate and things like that, but science is relatively consistent with this right now. We look at viruses and they don't meet the criteria to be called alive.